going to Eureka, bringing you the latest in scientific discoveries from my dorm room. Now, today's topic of discussion will be bioluminescence in sharks. Bioluminescence plays a critical role in the lives of deep-sea creatures, from camouflage to hunting. And while the light-producing mechanisms of bony fish are very well understood and have been for some time, virtually nothing is known about bioluminescence in sharks. And this is where we meet our two star scientists of the day. They're Julian Claes and Jerome Matterfeld, and they work for the Catholic University of Louvain in Belgium. They recently published a study in the Journal of Experimental Biology that details their analysis of the light-producing mechanisms in the velvet-bellied lantern shark, and they investigate what activates the photophores in their skin, which are the light-producing organs that give bioluminescent animals their glow. They use a special device called a luminometer to measure any kind of change in light production. And the reason that they chose neurotransmitters first is because bioluminescence in all the other animals that have been investigated so far has been triggered by the nervous system and nerve cells. However, all the neurotransmitters that they tried, adrenaline, gamma, didn't show any positive response, so they had to move on to something else. And they tried 3 hormones that are known to regulate skin coloration in sharks: melatonin, prolactin, and alpha-MSH. When they exposed these 3 into the shark's skin, it became very apparent that the photophores are controlled by these 3 hormones. When exposed to melatonin, the photophores gave off a dim, weak glow that lasted for a few hours. It's a long-term reaction. And the scientists explained that this would be better suited for countershading the organism to camouflage it from predators that may be looking for it. And when prolactin was injected into the skin of the sharks, they got a much stronger, brighter response from the photophores, but didn't last nearly as long. And they believe that this would be better suited for communication between individuals of the shark species. And finally, there was alpha-MSH, which inactivates the photophores and turns them off. Now, this is a significant find because this is the first ever example of hormones controlling bioluminescence in any animal. And it also provides evidence that bioluminescence in bony fish and sharks evolved separately under the same evolutionary pressures, which is a process known as convergent evolution. And the researchers believe that investigating other bioluminescent shark species will show that their photophores too are activated and inactivated by hormones. And that's all from me. Goodbye.